your textbook, open to page 207. Our finger is pointing to that page currently. I'd like you to write 10. 7, 6, left side, 5, 4, 207. Sit down, knees there, thank you. 3, 2, and 1. Here we go. I'd like you to look at the picture. See the picture spark. On page 207, there are 4 phases. We're going to walk through each one. Start us off. Read where it says A, prophase. The reader. Thank you. Yeah.
The chromatin forms to form visible chromosomes.
Well, I don't know what that means at all. So I'm going to go back into the text and actually read it. Go where it says prophase, the first phase of mitosis. I'm going to call on scholars to read. If you don't raise your hand, please be ready. Starting at the top, start us off, Keon.
On the prophase, the first and the longest phase of mitosis, the longest close of the 2 visible chromosomes.
Good. Charles.
Anaphase. As you can see in figure 8.12, duplicated chromosomes is made up of 2 halves. The 2 halves of the double structure are called sister
Chromatids.
sister chromatids, and the DNA they contain are exact copies of each other, and a form of DNA is copied during interphase.
Roger.
Sister chromatids are held together by a structure called centromere, which plays a role in chromosome movement during mitosis. By their characteristic location, centromeres also help scientists identify and study chromosomes.
Eyes here. So this is what I want you to get. I'm going to show you a little bit first, and then we'll talk about it. Please write this down. Next to prophase. DNA makes up this thing called genes. G-E-N-E-S. Genes, if you've heard of genetics, that's what genes are. They're just DNA.
Like when we do the tennis thing?
Exactly. Wonderful connection. Genes form what's called chromosomes. Chromosomes. Chromosomes look like an X. Please draw an X. Please make sure your head is up as we're learning. Good. Draw a dot in the center of the X. This whole thing is a chromosome. Each leg, each piece, is called a chromatid. Chromatid, with a D at the end. The center piece, this little ball right here, that's called a centromere. A centromere. So that's what holds the 2 sister chromatids, the 2 chromatids together. It's the centromere. So this is DNA. When you think of DNA, you always think of, oh, it's this really nice little helix. But it's not. DNA is like all over the place inside it's like everywhere. And during mitosis, during prophase, it makes nice little X's. Nice little chromosomes. Questions on it? What questions do you have?
No.
I see. So on the side, draw a cell. In the center, we're going to draw those chromosomes. They're kind of all over the place in the nucleus. We're going to draw the nucleus around it. The nucleus is broken right now. The nucleus is starting to dissolve as the cell prepares to divide. Okay. Repeat after me. Don't say anything yet. This X is called a chromosome. Chromosome. Say it.
Chromosome.
Good. Each leg is called a chromatid. Say it.
Chromatid.
The center is called a centromere. Say it.
Centromere.
All of that is made up of genes. Say genes.
Genes.
Which is made up of DNA. Say DNA.
DNA.
That's just kind of fun. What questions do you have about this? Ask your questions now. Track online.
You guys do this thing right now?
And we're going to watch a video first. Reading, video, and track with me. Track.
The centromere just keep all the chromatids in one.
The only purpose of the centromere is to hold the chromatids together and to break when they break. Darny.
How many, you know how you be saying chromosomes, like, never mind.
Go ahead, you're right.
I don't know how to say it. You know how they be saying chromosomes have a lot of, like, I don't know, never mind.
Your body has 21 chromosomes.
Yeah,
Sorry.
like, you know how you be saying that,
Your body doesn't have 21. Each cell has 21 chromosomes. Each chromosome has approximately 80,000 genes.
Are we writing this?
Nope. And every gene has about 1,600 nucleotides. So think about how many nucleotides, little individual pieces, nitrogenous bases, make up a single chromosome. It's huge. And then you have 100 trillion cells in your body. It's mind-boggling how many nitrogenous bases you have. Track chance.
What happens if you have more than 21?
Rewind.
Thank you. What happens when you have more than 21?
Your body. Not exactly sure. We'll get back to that. I was going to say, I don't think that you can, but I think there is a disease where you have more than 21 chromosomes. I don't remember the name right now. We'll get back. Track knowledge.
Like back to what she said about the Punnett squares, like, you know, like they have the X and the Y. Where are the Y's? Do they have one?
Yeah, I'm not drawing this right now, but a male chromosome, which is for men, half of our chromosomes are Y chromosomes, it actually isn't a full X. It looks like this. And it still has a centromere here. And it still breaks into two pieces. But it's actually a half a piece. They call this a Y chromosome because it kind of looks like, I don't know, kind of looks like a Y. So this is what male chromosomes look like. Males have pairs of these. Females only have the X's. It doesn't actually impact your protein development. It's just a sign of the sex that you are. Track ready.
Thank you. You said each cell have 21 chromosomes. So like when a cell divides, do the chromosomes divide?
I see her. Amazing. So each chromatid is actually a full strand of DNA. This is a full strand of DNA. This is also a full strand. Right? It replicated, created two of them. That was up here in, what was this phase right before mitosis? What comes before mitosis? Hands. What comes before mitosis? Trey? In interphase, one of the things that happens is DNA replicates. This is what DNA looks like when it replicates. Two different strands. And then it splits. So it starts out like this, it then replicates to this, and then it splits into those two. So you always have 21. Last question, Trey.
Thank you. What if you have like one of each, or either two are the same thing?
Sorry?
I said, what if you have one of each, an X and a Y, either two of the same?
And either two are the same? I'm sorry, I wasn't hearing.
Like one, okay, like you know how you say X is for females, Y is for males.
Females have two, females.
What if you have two X's and two Y's, either X and a Y?
Your body won't. So your body will always either have 2 X's, or will have 1 X and 1 Y. And it will never have 2 Y's, it will never have 3 X's, things like that. Okay, good. Maybe I'm misunderstanding, Charles. If I am, ask me. Go to metaphase, read letter B on the picture.
Right. Well, I was going to explain, like,
Pause. This is Mr. Twofer.
you can call me Mr. Max, it's
Mr.
fine.
Max.
Hi, everybody.
Oh, hold on a second. Try to mean. Mr. Max is actually here to, the reason the camera's there, he's actually here, a coach from Boston, who's specifically here to help myself and Ms. Wright, who got into a program to help us become better teachers. So what he does, this is actually, you shouldn't feel intimidated by the camera, although you guys are amazing. His job is to watch my teaching, give me feedback on tomorrow's lesson, so I can be better tomorrow. So the same way that you get feedback every day, I'm asking for feedback also. So this is Mr. Max.
Thank you. So, I'm sitting back here. I want to answer actually both of the questions that you have simultaneously. Mr. Pollack is a fantastic scientist, a great brain. You guys are very, very lucky to have him as a teacher. And I am able to answer these questions because I have the advantage of I'm hooked up to the internet right now, so I did a little search. So the answer is actually you can have an extra XY chromosome, and that creates a situation called Klinefelter's syndrome, which is what happens when you get that extra one, and that's the disease that he was referring to. So you can have that, and there is a disease or syndrome that forms as a result.
Okay, thank you. You said the male one, the male have like the will have X, and the women got the whole X. So Charles saying, what about somebody have both of those?
Yeah, is that your question, where you only have the halves, Charles?
That was part of my question. Like, I was wondering, he answered part, and the other part, no one answered because, like, my cousin, she worked in a nursing home, and she was telling me, I forgot the name that she said, it's a man. He's like a man, but he has like 2 parts.
Oh,
man
I
and
see
a
what
woman's
you-
part.
Alright.
And then, you know, because like I also know a lady that had a child, and he actually had 2. It was a male, and he had 2 of the same thing. So once he made a certain age, he had to get an operation to get one of them removed.
Yes, so
that's
there's a term called hermaphrodite. Hermaphrodite means that you have one of each. That is a genetic disorder. In general, one of them doesn't actually work. It's not like you have the plumbing hooked up to both. You have the plumbing hooked up to only one of them. So it's a surface surgery. It's not actually like going in and changing organs. It's a surface surgery. It is genetically related. I'm not exactly clear on how it happens. So let me ask you more questions. I apologize for not understanding your question. Go to letter B. All right. So just to be clear, we're about 3 minutes behind. I want to catch that time back up by having a really tight discussion for the rest of the time. Armando.
Metaphase. The chromosomes move to the equator of the spindle.
Spindle. Look at the picture. Tell me what equator means in this picture. Hands. It just means, Lauren.
The central?
Good. So write this down. Chromosomes move to center. Very nice. By the way, in prophase, I just need you to know this part. Prophase is where chromosomes are formed. Metaphase is where they move to the center. So the picture looks like this. They get lined up. The reason they get lined up is there are these things pulling on them. You can draw them in. Those are called, chance to hit your head up. These are called spindle fibers. The spindle fibers are pulling at the chromosomes, and that pulling is like a tug of war. They end up right in the middle. Okay. And they're going to pull even harder. That's going to cause anaphase. Track the paper. Track the book. Read letter C. Anaphase for now.
The centromere is split and the sister chromatids are pulled apart to opposite poles of the cell.
I see. So that's where the chromatids separate. Right? Chromatids are copies of each other. You end up with half on this side, half on this side of the cell. They've actually split. You've broken the chromosomes. The spindle fibers have pulled until they broke. Last one, letter D. Thank you, Lauren.
Telophase. Two distinct daughter cells are formed. The cells separate as the cell cycle proceeds into the next interphase.
So that's what I want you to write down is actually the two nuclei. So now in that one cell, I now have two separate nuclei. In that one cell, two nuclei are formed. With it.
Alright. You know, when you send out the mRNA, so you get like too much protein.
I'm sorry?
Are you gonna get too much protein?
If you have when?
Too
There's no protein synthesis happening right now. This is cell duplication. Once you get back to interphase, then you'll have protein synthesis. Is that your question?
Yeah.
Yeah. So protein synthesis won't happen during this time. Think about it. In order, I see it. In order for protein synthesis to happen, please try. The DNA has to separate. Right? That's how you get mRNA. In order to separate, it can't be in a chromosome. So you have to wait until you're deep into interphase. Hand. What comes after mitosis? That's incredible. What is cytokinesis? What is cytokinesis? I see a lot of hands up. Thank you, Trey.
Is it two cells separated?
It's after the telophase. Track, Tina. Eyes here. You now have two nuclei. The next step is when this pinches, creates two new cells. What questions do you have on mitosis? Armand.
How come they bring you
Meiosis has to do with meiosis. Meiosis has to do with the development of a sex cell, and the sex cell developing into one egg, into one cell. It's called gametes into zygotes. That has to do with reproduction. This is not sexual reproduction. This is when you cut yourself, how does it heal? This is how does a baby grow a brain? That's what this is. Track with me.
Alright. What if you
I
sell
love the tracking in the front, she's all.
what if you sell the prophase, but then you skip the telophase? Your eggs won't fall off?
So the metaphase is like, think about it, if I'm pulling on these three, I really wanted to do this. If I'm pulling on these three, the very first thing they're going to do is they're going to line up in the center. Right? Think about it. If I'm pulling on this side, and someone else is pulling on that side on ropes, the stuff in the middle is going to end up being pulled until it's in the center. And then you pull so hard, eventually it's going to break. Do you see that?
Yeah.
So actually, there's no way to skip phases here. This is basically them just being pulled into the ring.
Alright.
What you need to know, track here, form chromosomes. Move to center. Actually, repeat after me. Form chromosomes. Say it.
Form chromosomes.
Metaphase. Move to center.
Move to center.
Anaphase. Chromatids separate.
Chromatid separate.
Chromatid separate.
Chromatid separate.
Telophase. Two nuclei form.
Telophase form.
Good. I was a little bit unclear about that. That's okay. Eyes here. Close your books. Books put away. Right in front of you. 10 seconds. 10. 9. I don't want to lose any transition time. 7. 6. 5. 4. 3. 2. And 1. Start this board. I need a volunteer. Okay, as we're watching, Armani's going to read whatever comes up on the board. I want you to listen very carefully. Your job during this is to see how this relates to what you just learned. Be at level 0 so you can hear my question. It's just music in the background. I like it. It's kind of nice. These are cells in your body. This is actually what a real virus actually looks like and how it attacks your cells.
The earth is teeming with countless living cells.
This is an example of a neutron, which is what carries signals from your brain to the rest of your body. These are not actually videos
You have 100 trillion cells in your body alone.
of your actual cells. They can take pictures of them, but they can't take videos. It's too small.
There are you.
This is red blood cells, stuff that goes through your stream, your bloodstream. So cells don't all look the same, like little circles like I did before.
Cells come in all shapes and sizes.
They also grow in different ways. This is like the neurons that carry
They
information.
perform thousands of different functions in the life forms of Earth.
They fit together in different ways. This is showing you how they fit, how they separate. How they transfer from one area of the body to the other.
They work together to form the tissues of all living things.
So the red blood cells are cells, so is the tunnel. So is your heart. So is your brain. Everything in your body is cells except for bone. Bone is not made out of cells. Bone is made out of calcium.
But cells do not live forever.
Cells die all the time, and when you die, all of your cells die eventually.
Eventually they die.
The material that they give off, your body uses to make more cells.
Fortunately, they have the ability to reproduce first through a process called cell division.
That's what we do.
It begins with a chemical signal to prepare for reproduction. The cell grows larger. It's called the G1 phase of a cell's life. After growth, the cell duplicates its DNA. This is called the S phase of the cell cycle. The DNA is then checked by enzymes for mistakes and repair. This is the G2 phase of the cell cycle.
What is this part of the cell cycle called? Yes. We learned this as a different name. This whole thing is called. Love the enthusiasm. Thank you. Tina? This is interface. Good. So what do you think is going to come next? Class?
When red cell division begins, it is called mitosis.
Whoa, look at that. You're right.
And the cell divides.
Millions of them all the time. Here are the steps to mitosis. Please make sure you understand each step.
Mitosis begins when the nuclear membrane dissolves and the DNA thickens, called prophase.
Quick quiz. What is each of these X's called? I should say all hands up. Kayla. What's each piece of the chromosome called? Denzel. Chromatid? Good. What's the center of it called? Brunel.
Centromere.
What's it made out of?
Oh.
What's it made out of? Darn. What's it made out of? Leon? Before that, Armani. Genes which are made out of DNA. Please continue. So this is prophase. It breaks open and it forms chromosomes.
Special fibers called spindle fibers from on each end of the cell and grow to
The DNA chromosomes. I'm sorry. So these are those ropes.
The fibers then attach to centromeres on the chromosomes and pull them into a line in the middle of the cell called metaphase.
I want you actually to look at the next picture. This is my favorite picture of metaphase.
The chromosomes.
Thoughts? This, imagine a cell. It's not actually a circle, it's a sphere. And they get lined up across the hemisphere, like the equator. So that's what's going on. They're all across the middle. Sorry.
The chromosomes are pulled apart by the spindle fibers to opposite sides of the cell coin.
So they actually get broken apart. Oh, it's pulling. Great.
Last is telophase. Two nuclei in the cell then divide in two called kinesis.
Cytokinesis. Eyes here. So you can see the two nuclei forming. And that's telophase, and then they break into two. That's cytokinesis.
All of us started life as a single cell that divided again and again.
So this, believe it or not, is the very first picture that you ever were. This is who you were in the very, very beginning.
Embryo.
All right. Eyes here. Questions? Track Lauren.
you
Pause.
know-
I'll check your spark. We just watched a video. Turn to Lauren. Right.
You know how when the like when they be finding dead bodies, and you said the cells, something about the cells dying. The cells can't die before you die.
So they can. That's what, like, okay. Sometimes when you go to the hospital, you see people who are like bedridden. They can't get out of bed anymore. You can't move and they like don't have good circulation. And if you look at their feet have become like blackened. Like then they start to peel and they start to smell really bad. And what's happening is their cells can't get oxygen because the blood doesn't go to their legs. And their legs actually start to die, even though they're still alive. Okay, so answer is yes. 2, when you die, your cells live a little longer. So your hair actually continues to grow after you're dead. It does. Your fingernails continue to grow after you die. But that's not actually life, that's cell replication. Sorry.
How your fingernails grow in your skeleton?
No, you're not a skeleton yet. If someone cut my head off right now and I fell on the floor, a week later I wouldn't be a skeleton yet. I have no idea. It takes many years to become a skeleton. 2 questions, I want to move forward. Chance.
Yeah, my grandpa.
Rewind. Start with thank you.
Thank you.
You're welcome.
My great grandpa. I think 3 or 4 days before he died, which was like a couple weeks ago, he like all his legs, like on the side of him, it was like black, and on his hands, like everything was dark and stuff. And the only thing that really was kind of like brown and looked like it was full of life was like everything like by his chest. Other than that, he couldn't get any blood to it.
Your body knows how to prioritize. If it doesn't have enough blood or blood pressure to get to your fingers, it knows you can live without your fingers, but you can't live without your heart. So your fingers will die first in that case. Come on.
So like say if you shave all this off and then like you die, and you know how when it grow back like a week later, it's gonna be growing back?
Absolutely.
What?
Absolutely. If I went clean, which hasn't been in a long time, if I clean shaved totally, and then I died, and you dug me up a week later, I would have a nice sized beard, as if I was still alive. So that's something to think about. I don't know. Whatever. Turn to your red sheet, listen for directions, track me. Here's what's going to happen. I need your full focus right here. In the cups, there's some around your desk, and I'll deal with the who has cups in a second. In there are scissors. Track here, scissors. There's glue. And there are these little beads called centromeres. These are going to be your centromeres. You also have what looks like a pipe cleaner, but is actually chromatin. You're going to take your chromatin and you're going to cut it into pieces, because we used to fit on this paper, and you're going to put it through the centromere, and all of a sudden, you're going to take.
Yeah, like we just found out about
You're going
fire.
to take. Are you listening? You're going to take these chromosomes, at least 2, and you're going to scatter them for prophase. You're going to line them up for metaphase, and then for anaphase, you're going to break them apart on opposite sides. And then for telophase, you're going to break them apart and draw nuclei around them. Now we are a good 5 minutes later than the other classes. I do want you to have time to do this. I think it's important for you to get the feeling of your hands on some chromatids and some centromeres. 1, 2, 3, 4, 5, 6. I'd like a group in the back to form. Move your desk if you have to. I want you into lab groups and working in the next 12 seconds. Go. 12. 11. 3 of you. Now. 6. 5. 4. 3. 2. And 1. You have 8 minutes.
Making the things.
You get as many as you need. Two pre-start.
Mama is not small.
What?
Why is not
Yeah, you got a small one.